Crime scene investigators often find small amounts of DNA at a crime scene. A small amount of DNA is not enough to analyze on its own. Forensic scientists use PCR or polymerase chain reaction to amplify the DNA, which means to make millions of copies of the DNA. So they do this using an enzyme called DNA polymerase, nucleotides, which have our nitrogenous bases, our A, C, T, and G, and a set of primers. In the test tube, there will be the target DNA. These polymerase, nucleotides, and primers and the target DNA are all going to be added together. The temperature is going to be raised, and that's going to cause the strands to separate. We want them apart so that we can replicate them. The primers are then, after the temperature is cold, the primers are going to then bind to the sticky ends of the DNA. And then DNA polymerase is going to go down the strand of DNA, synthesizing a copy of the DNA. So after the first round of PCR, we now have 2 copies of the DNA, and those 2 copies are going to go through PCR again, and those 2 copies will produce 4 copies, and those 4 copies will produce 8 copies, and this will keep going until there's millions of copies of the DNA. So in approximately 40 minutes, researchers can take one sequence of DNA and make millions of copies. They can take the DNA from the crime scene and compare it to suspects that they think may have done the crime. They extract the DNA from those suspects and run a comparison electrophoresis analysis to see which banding patterns match the guilty party. Because humans' sequences differ slightly, we can use DNA profiling to compare the DNA found at a crime scene to our suspects and hopefully find the one who committed the crime. There have been many cases that people have been exonerated because DNA was found, and now that we have this, they have been tested and were proved not to have done the crime that they're doing the time for.